Well, hello again, and in this video, video number 7 for chapter 3, we are looking at the cell cycle. This is getting away from, we just have been looking at movements in and out of cells and the different parts of it. Now we're going to look at how cells, normal cell life, the cell cycle. So the cell cycle includes both normal life processes, which we're going to say is interphase, and then division. There's two types of division. Mitosis is the division of the nucleus and the genetic material. Cytokinesis is the division of the cytoplasm, so everything else. At the end of mitosis, understand that the two daughter cells will have identical genetic material. The DNA and stuff, this is identical. So of the two, when it's done, this cytokinesis, the splitting of the cytoplasm, which is the fluid and the organelles, is not even. And so when we look at this, we're going to talk about what's happening in the first part of interphase, the normal cell life, because it's all about making sure that we start rectifying the not even part of, gosh almighty, that's terrible, not even part of this cytokinesis. I'm having trouble with my mouse right now. But anyway, when I'm going through this, most of the time I find this is review for my students, but far more than the chemistry stuff, but it's all right if it's not. So this is a little picture that shows the cell cycle. And again, right here, this is at the end. We've got two different daughter cells here. And so if we take the one and start, take this one away, it's got its own cycle. This one comes in and it starts with the G1 phase. This is cell growth. And we're going to look at what this is doing is basically inventory. It's when the cell is going to look to see what organelles it has. Does it have too much of some sort of an organelle or not enough? And it starts breaking down those that we have too much and creating the ones that we don't have enough of. Because again, remember, at the end of the division, in mitosis, we have two daughter cells with identical genetic material, but different everything else. So G1 phase is when the cell is doing the act of inventory. Then it goes into this S phase. In the S phase, we are going to replicate DNA, which just means I'm going to make a copy of it. So I'm going to take this DNA strand, this huge molecule, I'm going to basically rip it in half and make copies of it. So I'm going to have two, at the end of the S phase, I'm going to have two identical DNA strands inside the nucleus. Then we go into G2 phase, and then pretty quickly we end up in what we call mitosis. Now, mitosis is what we're going to look at. There are 4 phases. If you include interphase in the cell cycle, you can remember that it is I passed my anatomy test. Now there are other ones, but I kind of like that one. It's pretty straightforward and pretty clean and hopefully true. So interphase, like we said, is the period of normal cell life. It's where we're going to replicate the DNA. It's going to synthesize, it's going to make organelles. It'll break down ones that we have too many of. And the big important one for us is this S phase, because the S phase is where DNA is replicated, and that is an easy low-hanging fruit type question. That is one that I would guarantee you that you will see. Now when we look at mitosis, again, two different things. Mitosis is only about the nucleus. Cytokinesis is everything else, all right? So we're just going to kind of talk about mitosis right now. Mitosis has 4 phases. Like we said, prophase, metaphase, anaphase, and telophase. So passed my anatomy test. Prophase and telophase are kind of long processes. Metaphase is a quick snapshot. Anaphase is a little bit longer, but it is also a very short apart. So prophase, I'm going to have a lot of things that are going to unravel, things that are going to kind of disappear, and you're going to see those chromosomes, the chromatin will start coalescing and forming those familiar X chromosomes, and they'll start working their way towards what we'll call the equator. There's other things that are going down. The nuclear envelope dissolves. You've got centrioles will move to opposite sides. But as that happens, the chromosomes will line up in the middle, and once they're in the middle, we call that metaphase. This is a very, very short time. As soon as they get in the middle, you can say the chromosomes line up in the middle, boom, they're going to split and start being pulled to different sides. At that point, you're in anaphase. Now, one of the things about anaphase to understand, anaphase, again, when things start getting pulled apart, that's also where cytokinesis starts. And so anaphase, I'm going to start pulling the genetic material from side to side, as well as the cytoplasm will start doing it. But really quickly, you'll start seeing all the things that were getting pulled apart or disappearing in prophase start getting put back together. And so we will end up pretty quickly getting into what we call telophase. And in telophase, basically all the things that were, again, going on in prophase are being reversed, and the cell is being pulled apart. So we'll see a couple pictures of this. So here's the common picture, the picture that is in the 112 book, and you kind of see it in different things. This is the cycle. So this is the cycle of mitosis. So we start here. Again, this is prophase. Again, it could be late interphase. But what would happen if you were just bored and had a microscope and decided I just want to watch some cells? You'd be looking at them like, hey, cool, and then all of a sudden you'd start seeing a change. You'd start, if you were really able to see, if you had a good microscope, you would see that the centrioles, the centries would start moving. And then you'd notice that instead of just this random kind of darkened dot that you had been watching, you'll start seeing that there's some darkened spots that will start appearing. You can kind of see them on this picture. That's going to trigger you to kind of think, hey, something's going on. And as you continue to watch, you'll see that the nuclear envelope will start disappearing. You see it in this picture over here. There's no little envelope anymore, and the chromosomes now are not just this general dark area. Now you actually see the individual parts to them. And those are chromosomes. Now this again is prophase, and as you start watching, these chromosomes will start spreading out and trying to line up along the center. Now again, we're still in prophase. But if I continue to watch, they will eventually line up right along the equator. Boom. And at this point, this snapshot is what we call metaphase. It does not last long. As soon as they get there, they're going to snap and then be broken in half and start pulling from side to side. And as soon as that happens, we know we are in anaphase. Again, good picture of it right here. Now, as I am watching this, what's going to happen is these little areas where the chromosomes are, the little halves of chromosomes, will start kind of grouping together. We'll start seeing the nuclear envelope starting to reform. We'll start seeing what we call a cleavage furrow, and we'll start kind of noticing all the things that were getting taken apart over here in prophase are now going to be being put back together over here in what we call telophase. So the beginning and the end are long kind of processes. The two in the middle are pretty short, but in particular, metaphase is very short. So those are the stages of it. I hope that kind of makes sense and that you're kind of familiar with it. Now, cytokinesis, again, cytokinesis begins in anaphase. So remember that once those little chromosomes line up in the middle and they start getting pulled, they line up in the middle, metaphase, and then they get pulled apart from side to side. As soon as they start getting pulled apart, everything starts getting pulled apart. And so cytokinesis, which is the division of the cytoplasm, begins during anaphase. Right? And it's going to continue through telophase. Once it starts, everything is going to be divided together. And so when the new cell is formed, it's going to have identical DNA, but it's going to have different things like size and organelles. This is showing it kind of, to me, this just looks like, this almost looks like a student when you get told they forgot that there's a test or something, and you get told there's a test, and they kind of have that look. Anyway, sorry, just a little joke. But this is kind of showing an actual electron microscope picture of this as it's dividing. Now, the mitotic clock, the idea of how often cells go through mitosis is different for different cells. But the thing is, on the tip of the chromosomes, there is a special thing called a telomere. Every time, so if I have a chromosome like this, and I'm just, all right, so draw, all right, at the tip of these little chromatids things, right at the top here, I'm going to have this thing called a telomere. And every time it divides, a little bit of it gets taken away. All right. And so it's a built-in countdown timer. Once it's gone, the cell can no longer grow. It can no longer go through mitosis. And it's kind of a self-preservation, because if things can continue to go through mitosis, they have a tendency to mutate and have issues. So as they're doing this, it's going to go through this process of mitosis, and the telomere gets shortened every time until it no longer is there, and that cell cannot go through mitosis, and it will end up dying. Now, cell division is controlled not only by the type of cell it is, but it's controlled by hormones. Obviously, human growth hormone, or HGH, is going to cause cells to grow. There are growth factors that are released. You'll see that when we study about cuts, you'll see that when a cut happens, there's going to be a growth hormone, a growth factor that's released and causes the cells to start growing. And we have what's called contact inhibition, which just basically means cells are going to grow to fill the space they're in. So if you're sitting in a room, and imagine there's a couple of you, but you are cells, you would have the ability to make new copies of yourself until that room was filled and you could not grow anymore. And they would call that contact inhibition. Now, when we talk about tumors, we're not going to get really in depth in this. I want you to know the difference between benign and malignant. Benign tumors remain local. While it might enlarge, they are not considered life-threatening. Malignant, on the other hand, spread, which we call metastasize, and they are invasive, and both of them are cancerous, but these are true cancerous cells. These are the deadly ones, malignant. Now, mutations will occur in the genes that cause cancer, and again, not worried about these oncogenes. Now, if you know an oncologist is someone that is about cancer, it kind of makes sense that these are the genes. And then we've got tumor suppressing genes, but they end up getting inactivated. They kind of get muted, and so mitosis is no longer regulated, and you'll see this in the next little list. So this is a picture of them. These are talking about the cancer cells. Again, not going to ask you about this. But this little list of the characteristics of cancer cells is pretty, you'll see that most of it is kind of the same thing. Invasiveness, ability to metastasize, it spreads, which means it's invasive. It's lost its contact inhibition, which means it's just going to keep making copies of itself. It all boils down to loss of control of the cell cycle. Now, the other thing that I think is a pretty important thing to understand or kind of know is cancers have the ability to hijack our cardiovascular system. It causes our body to create a special capillary bed just for that area of cancer cells. And that's one of the big areas we are learning to fight cancer in, is trying to stop this, because basically what it's doing is it's almost like tapping into the energy source. And so it has a direct blood supply and can end up causing it to be very invasive, where it spreads like crazy and grows like crazy because it's got a direct blood supply. It's drinking straight from the garden hose. Now, again, just a picture showing how it kind of in this, again, showing how it's taken and hijacking the capillaries to make it happen. But again, as far as cancer goes, I want you to know in this class, all we're looking at is benign versus malignant. and understanding that the malignant type can really spread all over because they lose control of their mitotic clock. They just start reproducing, they're not specialized, and they take over the capillaries, the cardiovascular system by tapping and forcing it to make a direct capillary bed to feed it. And so it's like this overgrown monster. But anyway, I hope all of that makes sense. Again, I hope the cell cycle makes sense. That's the most important part in this as far as our class goes. Knowing the different parts of it, knowing interphase and then mitosis and the difference between mitosis and cytokinesis and when cytokinesis starts. So I hope that all makes sense. Have a wonderful day.